Chapter 1. Now, for those of you who haven't taken a class in this format before, once I go into presentation mode, I can't see the chat box. So if you want to ask a question, please unmute yourself and go ahead right away and interrupt me. That's fine. I don't mind at all being interrupted. I just, I can't see the chat box, and I know some people ask questions there, and then they don't realize that I can't see that. So I'm going to hit presentation mode, and I won't be able to see that. So feel free to interrupt. All right, here we go. Chapter 1 of microbiology, Introduction to Microbiology. So microorganisms, they are defined as organisms that are too small to be seen with the unaided eye. A germ refers to a rapidly growing cell. A germ is not necessarily pathogenic. It very specifically says it's a rapidly growing cell. Oops, sorry. So any microbe can be a germ because they do divide rapidly. When we talk about pathogenic organisms, there are actually very few that are disease-causing pathogenic microbes. Most are good microbes. They decompose organic waste. That's what we're looking at here. These are some pools from a water treatment plant. They are producers in the ecosystem by photosynthesis. An enormous amount of photosynthesis is conducted by microbes. We always think of trees, but in fact, there are more microbes than there are plants. And the microbes are just adding so much by way of photosynthesis, by fixing carbon, by releasing oxygen, by taking that CO2 out of the atmosphere. We use microbes to produce industrial chemicals. Some examples are ethanol and acetone. And they also, of course, produce food. So we can do fermentation for foods such as vinegar, cheese, bread, soy sauce. They also produce other products that are used in manufacturing, such as the material cellulase, and also in disease treatment. So we can use biotechnology to have microbes, trick them into using a human gene and expressing a human gene to make something such as insulin. So the knowledge of microorganisms have allowed humans to prevent food spoilage. And what we're looking at here, sorry, I caught my daughter's cold. Now my nose is all runny. Summer colds are fun. What we're looking at here in this picture is pasteurization. And there are two ways to pasteurize raw milk. One is at low temperature, which is a low temperature for a long time, or the other method is high temperature for a short time. In the United States, we use the low temperature, which is why our milk has to be refrigerated and has an expiration date, a very specific expiration date on it. The other method, the high temperature, is closer to sterilization of the milk, and therefore it can be stored on the shelf. And that's what they do in Europe. I would argue it doesn't taste as good, but that's just something. Pasteurization at that low temperature does not kill everything. It just kills most of the bacteria and microbes in there. Our knowledge of microbes allows us to prevent disease occurrence. So Lister was really the guy that told us, wash your hands. So that led to aseptic techniques to prevent contamination in medicine and also in our laboratories. Very often in our laboratories, we're protecting the experiment, not ourselves. We're wearing gloves and dealing with our experiments, we want to be very aseptic. So the naming and classification of organisms. Carolus Linnaeus was the namer of all of the organisms. Well, he's the one who started it, let's put it that way. He established the system of scientific nomenclature. Each organism has two names, the genus and specific epithet. Whoops. So I wanted to point out this figure. We start with the domain. In this case, they're looking at the eukarya domain, which then goes down to the kingdom, phylum, class, order, family, genus, and species. We like to call it in microbiology the specific epitaph, not the species, because we're talking about microorganisms, not animals. Scientific names. Scientific names are italicized or underlined. The genus is capitalized and the specific epitaph is lowercase. This is important. We use this when you italicize, we know that it's an official name, that it has been accepted by the scientific community. That is the name that is published. If you don't use this specific capitalization and italices, then you might assume that it's a common name. So like, for example, an E. coli infection. So say you have stomach E. coli is going to be your scientific name. You're throwing up, you're sick. That's what's going to be put on the all the medical records. But what you might say to a friend is, I have a stomach flu. Well, it's not flu at all. Actually, flu is a completely different microbe, but that's not the point. The point is that you might use this very common name. I've got a 24-hour flu. I've got the stomach flu. And that's a common name that is inaccurate and will not be put on any medical record. So it is extremely important to note this, that you do, if you're using a specific name, that you are italicizing it. Now, why we can also use underlined is because if you're handwriting it, it's hard to tell if you've italicized something. So if you're handwriting an organism, underline it for its specific name. Scientific names are also Latinized, and these are used worldwide. They may be descriptive to honor a scientist, such as E. coli. So Escherichia coli honors the discoverer Theodore Escherichia, and it also describes the bacterium's habitat, the large intestine or colon. So we have Escherichia for his name and coli for where its habitat is. Another example is Staphylococcus aureus. Staphylo describes clusters. Spherical is cocci, which is the circles. And then the aureus describes the gold color that we see here. Now, scientific names after the first use, the scientific name may be abbreviated with the first letter of the genus and the specific epitaph. So we might write the first sentence, Escherichia coli and Staphylococcus aureus are found in the human body. Your next sentence would be E. coli is found in the large intestine and S. aureus is on the skin. So these are appropriate names. The first time that you see them, please write the entire name. The second time, you may shorten it. So the different types of organisms. We have bacteria, archaea, fungi, protozoa, algae, and viruses. And we're going to go through all of these briefly. We do have full chapters, they're modified because we are using a new book here, but we do have full chapters that will go into each of these individually as well. So this is, keep in mind, just a brief introduction. Oh, sorry, I forgot the multicellular animal parasites. I always forget that one. And the reason we have multicellular animal parasites is because they do have a microscopic part of their life cycle. So we'll start with bacteria. Bacteria are prokaryotes. Now, if you remember prokaryotes from your biology class, prokaryotes do not have any organelles. Bacteria also contain peptidoglycan cell walls. There are two types of that peptidoglycan cell wall, gram-positive and gram-negative. Gram-positive has far more of this peptidoglycan, whereas the gram-negative has a lipid bilayer with the peptidoglycan, and so it's more slippery, I'd say. But they both do contain peptidoglycan. Bacteria divide by binary fission, which is one of the reasons they can divide and multiply so quickly. That's where one cell simply splits into two identical cells. For energy, bacteria can use organic chemicals, inorganic chemicals, or photosynthesis. So it completely depends on the type of bacteria that you're talking about. Archaea. Archaea are prokaryotic. Again, no organelles. Archaea lack peptidoglycan. Instead, they have muric acid cell walls, so they're going to stain acid fast, not a gram stain. They tend to live in extreme environments, which is why we have the picture of Yellowstone Park here. And they include, these extreme environments include methanogens. So these are organisms that are going to use methane. Extreme halophiles. These are microorganisms that prefer an environment that's high in salt or sugar, but really it's salt because we don't have sugar out in nature. And extreme thermophiles, meaning that they like it very, very hot. Fungi. Fungi are eukaryotes. They contain chitin in their cell walls. Chitin is a very interesting component. We find chitin also in the exoskeleton of insects and in shellfish, such as shrimp. So if you're allergic to something like shrimp, you'll probably likely also have an allergic reaction to accidentally eating a bug. I don't know how that would happen, but if you did, and you probably would have a stronger allergic reaction to any fungal infection because of that chitin. Fungi use organic chemicals for energy. Oh, I forgot to say these are eukaryotes. I mean, I did say it, but I didn't say it. Eukaryotes have enclosed organelles. So they have a nucleus. They have an endoplasmic reticulum. So there are multicellular fungi. Those are molds and mushrooms. Masses of mycelia. And these can be composed of filaments or hyphae. And that's kind of what we're seeing here. We see some hyphae in this picture. It's a bread mold where they're sticking up and they have the little kinetospore at the top. Yeasts are a type of fungi and they are all unicellular. Excuse me. Protozoa. Protozoa are microbes that are eukaryotes. They absorb or ingest organic chemicals. And they may be motile. And they might use a pseudopod, which means a sort of foot, cilia, which are little hair-like projections around, or flagella, which are a little bit more tail-like. I wanted to say one more thing about protozoa. We find protozoa in the kingdom Animalia. It's the only one in the Animalia that would be a true microbe. Again, we do have those multicellular animal parasites. They're not true microbes. They are just included in microbiology because they have a life cycle stage where it's too small to be seen without a microscope. Algae are also eukaryotes. They contain cellulose cell walls, and they use photosynthesis for energy. So obviously we find a lot of algae in water sources. One of the good things about algae is that they produce molecular oxygen and organic compounds. One of the bad things is if you have too much algae in a particular water system, it can choke the life that's there. Usually we find this with really high nitrogen compounds in the water. So anywhere you're downstream from a water treatment plant, you want to test for nitrates because that could increase algae growth. Viruses. Viruses are extremely unique. These are acellular. And what I mean by that is that they're not actually really alive. They don't have metabolism. They don't create energy. And they cannot divide on their own. They consist of either a DNA or an RNA core. They don't have both, and you need both in order to multiply. So this is why they need a host cell. A viral core is surrounded by a protein coat, and that coat may be enclosed in a lipid envelope. That lipid envelope is usually derived from the membrane of a host cell. Viruses will only replicate when they are in a living host cell. They cannot replicate outside of a host cell, and they cannot do any metabolic processes. This is why they are in a category of their own. So these multicellular animal parasites that we talk about, they are eukaryotes. They're multicellular animals. These can include the parasitic flatworms and roundworms. Those are called helminths. So anytime you see the word helminths, that means worm. The reason they are included in microbiology is because they have a microscopic stage in their life cycle. That's usually the egg stage. It's very small. So the classification of microorganisms. There are 3 domains: bacteria, archaea, and eukarya. When we look at each one of these, bacteria are the oldest. So if you look down at the base of the tree there, we see that bacteria is the very first, or an ancestor of bacteria. It's the first to derive on our planet. And then we have the development of archaea and eukarya following that. These arrows are indicating a cross-transfer of genes in which we have the origin of mitochondria and the origin of chloroplasts. In eukarya, we have protists, fungi, plants, and animals. The only 2 that are going to be microbes will be those protists and fungi. We're not going to include, obviously, plants and animals as microorganisms. So we're going to give a brief history of microbiology. So the ancestors of bacteria were the first life on Earth. And that's, I underlined that there, bacteria were the first. However, the first microbes were not observed until 1673. Oops, yeah. Prior to that first observation in 1673, Robert Hooke in 1665, he reported that living things are composed of little boxes or cells. In 1858, we have Virchow saying that cells arise from pre-existing cells. We can't have things spontaneously come to life. The cell theory is that all living things are composed of cells and come from pre-existing cells. In 1673, we have Anton van Leeuwenhoek. He is the first one to actually start to realize. Oh, sorry, yes?
Yeah. Can you slow down a little bit?
Oh, sorry.
That's okay. And then quick question. I don't remember if this was mentioned, but are these lectures on Brightspace?
I'm going to try to upload them.
Okay.
I don't know yet.
Okay, thank you.
But PowerPoint is for sure. So you'll have this whole PowerPoint that you can click through on your own.
Okay.
I just don't know if it's going to upload this whole talk.
That's fine. Yeah, as long as the lectures are there. Well, thanks.
Okay. Sorry. Just get into this too much. Okay. So the first observations, we're going back to 1673. We have Anton van Leeuwenhoek, and he's describing live microorganisms. The way that he was describing them really had to do with what he could see. This is his microscope. I'm pretty sure everyone in here has seen a microscope, even if you've never used one before. They're a lot bigger than this. This is literally, what is this, 9 centimeters? It's very, very small. And the lens is at the very top there, and you have this little pin, and on top of that pin, he'd put a drop, tiny little drop of a microorganism, and then he would hold it and look through the lens at the microbe. So you can imagine if you don't have a very steady hand, it's hard to see. And also, that lens is going to be specific to somebody who has the same sight. So, they didn't have contact lenses back then. They didn't really have very good glasses either. So a lot of people didn't believe Anton van Leeuwenhoek. They thought he was making this up. Some people did believe him. Some people were able to see the microorganisms. But he also really kept this very secret how he made his microscopes. So his lens was his own creation. And so, well, for better or worse, he really had a patent on it. And he drew a lot of these microorganisms and he published a lot of papers, but he couldn't take pictures of them, obviously. And there were a lot of people that couldn't see what he saw. And again, his microscope couldn't be replicated. So it was quite a while, actually, before people even believed that microorganisms existed. So if we look at his lifespan here, 1673 to 1723, we have Anton van Leeuwenhoek. Let's go back a slide. We don't have until 1858. So that's what, almost 100 years later, where Virchow starts to say that cells arise from pre-existing cells, and we have the cell theory. So even though Leeuwenhoek had visualized microbes, most of the world didn't believe him, and it wasn't even posited that pre-existing cells had to exist for cell theory until 1858. And even then, it was a fight. So this is where the fight is. The debate over spontaneous generation. Spontaneous generation is the hypothesis that living organisms arise from non-living matter, where a vital force forms life. Now, this might make you laugh a little bit. You think that's just silly. But if you can't see microorganisms, if you can't see that microbe state of the multicellular animal parasite, you just see a worm all of a sudden appear, you're going to probably take some faith in this. I mean, people at that time thought that flies were created from meat and that frogs came from mud, which is interesting, but they weren't seeing the eggs that were laid. Now, biogenesis is the cell theory, which is the hypothesis that living organisms arise from pre-existing life. So obviously, biogenesis is the correct theory. Well, not theory. It is correct. Spontaneous generation is not correct. But again, we're looking at what people could even observe. So how could this be proven? The theory of biogenesis was not finally proved until we have Louis Pasteur come along. So we had Virchow say cells can only come from other cells in 1885, but it really wasn't until Louis Pasteur later in his life, and you can see his lifespan goes until 1895, was finally able to really prove that cell theory, prove biogenesis. And he did this using an S-shaped flask, where this S-shaped flask kept microbes out but let air in. If you had a flask, you can imagine having just a flask, and you sterilize it, you boil everything out of it, and then you plug it up. Even if you were to introduce a microbe in there before you plug it up, it's not necessarily going to be able to grow unless it's an anaerobic microbe. And they didn't know the difference between anaerobic and aerobic, things that lived without oxygen, that lived with oxygen. Not to mention, it's very difficult to culture, even today, an anaerobic microorganism. So they had this problem where they couldn't get anything to grow. So then what he did is he created this S-shaped, where the microbes weren't able to migrate into the flask, but air, being very small particles, still could get into the flask. So he boiled his media so it was sterile. He introduced his microbe. He put on the S-shaped flask, or the S shape of it, and then whatever he had introduced in there could grow. And then he would do this in tandem with something where he hadn't introduced any cells, and that wouldn't have anything grow at all. And so Pasteur was finally able to put to rest this debate that no, we don't have things spontaneously arising. They must arise from a previous cell. And this led to then the golden age of microbiology. So beginning with Pasteur's work, we were able to start to discover relationships between microbes and disease, immunity, and antimicrobial drugs. So Pasteur was also able to show that microbes are responsible for fermentation. So in this way, he was actually starting to get close to those anaerobic microbes. Do I have another thing? I do. Fermentation is scientifically defined. Well, it's not scientific. It is defined as the conversion of sugar to alcohol to make beer and wine. It can also convert other things, such as to make cheese. Microbial growth, as we know, is also responsible for the spoilage of food. Now, prior to Pasteur's work, people did know that they had to keep their food from spoiling. So they would salt it, they would dehydrate it, they would put it in high vinegar, so pickle it, right? So people knew that they had to, that food would spoil, but they didn't know that microbial growth was responsible for the spoilage of food. And so we have Pasteur saying, I know that microbes are causing part of this. Let's use a technique that I'm going to call pasteurization, and we'll use heat to kill off most of these microbes for a short time, and then we can prevent spoilage. So for your milk, instead of having to either drink it right away or, which you shouldn't do, don't drink raw milk. We'll go through that. We have other presentations that will show you how many different very dangerous microbes there actually are. Just heat it up, just pasteurize it. Just below boiling for like 40 seconds will do it. Anyway, you don't want to add a bunch of salt to your milk to preserve it or anything like that. So he was able to show pasteurization can also lead to the reduction of microbes. I think that's everything on this slide. Okay, so moving on to the germ theory of disease. So in the 1860s, so we're talking around the time of the Civil War here in America. And if you've ever read anything about the Civil War or seen any movies or even, some jokes, some skits on this stuff, they chopped off a lot of limbs. They didn't really do this very sterilely. They just kind of walked around with a saw and cut off the limb that had been blown apart. And the surgeons didn't wear gloves. They didn't wash their hands. They just took their little surgeon kit from person to person. So around this time, Joseph Lister applied Pasteur's work. He said, there are microbes in the air. These microbes can spoil food. They can cause animal diseases. I think they can also cause human diseases. And so he started introducing chemical disinfectants to prevent surgical wound infections. And they all thought he was a little bit crazy at the time. He was just using iodine. And then, well, in this case, they have phenol here, but iodine is a phenol. But he used iodine a lot. And it was Lister who really started the aseptic techniques. And we do still have some things named after him, including Listerine, the mouthwash. So if you ever think about how dirty your mouth is, it is quite dirty. And so you can aseptically clean it with Listerine. And you can thank Joseph Lister for that. So Robert Koch, he took this a step further. So he didn't just say, let's clean up our area, let's clean our instruments. He said, I'm going to prove that a microbe that infects an animal or a human can be taken out of that animal or human and recultured so that I can tell what specific microbe is causing a disease. And he did this using anthrax. So he provided some experimental steps called Koch's postulates. And these postulates were there to prove that a specific microbe causes specific disease. Oops. I thought I had his postulates on this, but I think I have it in a different presentation. Sorry, like I said, I'm switching to a new book. I've only had a week to look at this material. It's similar, but a little different, so I apologize. But anyway, what Koch's postulates, and we will see, I'm pretty sure we'll still have this figure in there, is that he would have anthrax, and he would give it to a mouse, poor little mouse, would then die. And then he would get sick first and then die, and he'd write all his notes. And then he'd take that mouse and he would take from its bloodstream, he was able to re-isolate anthrax. So he could prove that anthrax was the cause of death all along the way. Excuse me, I'm sorry. Something's going on in the daycares again. All right, so let's go on to vaccination. Now, vaccination predates a lot of this speculation on spontaneous generation and biogenesis. Like I said, people, while they couldn't see microbes, people weren't exactly stupid, They knew that they had to prevent spoilage of food. And they also, through just observations, could see if somebody got ill and recovered from that they usually wouldn't get ill again from the same disease. This was huge, actually, during the plague, which was back in the 1300s. That would wipe out entire towns. There'd be like one person that would survive, and that one person would then go to all these other towns that were ravaged by the plague, and they would still survive, and they ended up being the nurses of the time, or helping out other people. So it was observed that somebody survives a disease, they will not catch it again. And so Edward Jenner in 1796 really came up with this idea of vaccination. And he inoculated a person with cowpox virus. And that person was then protected from smallpox. Well, the true story is that he was a doctor, and he already had observed this sort of thing, but he didn't know exactly how it worked. And he had people come into his office, and he had a lady come in one day, and she was a milkmaid. And he said, "What's on your hands?" And she had cowpox on her hands. She said, "Oh, it's just cowpox. You get it from milking cows." And he thought about that, and he looked at those. He said, "Those look really similar to smallpox." And then what he did is he took the cowpox pustules, and he then used those to create his vaccine. And he inoculated a young boy, actually, who was then protected from smallpox. And then they went around just inoculating, whole schools and things like this. They didn't exactly have proper paperwork. They just went around and said, "Let's see if this will work for you." And it was a long time ago. Vaccination is derived from vacca, for cows. So that goes back to, it really started with this cowpox virus. I should put a picture of Jenner on this slide, because, he was really kind of a visionary for his time. And Jenner went around the entire world. He got on every ship he could. He was British, and so at the time they had quite a fleet. And his vision was to rid the world of smallpox, and which was pretty ambitious. In 1976 was the last case of smallpox on the planet. And that case, of the last, I don't know, I want to say natural case. And I believe it was in India. I believe it was in India. There were two other people that caught it in 1978 and I think in 79, and they were people who were handling it in the lab. But because of vaccination, we were able to eradicate smallpox from the planet. And there's a very small vial of smallpox that still exists, and that is in a frozen, hidden laboratory somewhere in the Netherlands. The protection that we get from vaccination is called immunity. And again, that's, you can't recatch the disease. Okay, so let's move on to treatments. So vaccines are preventative for the most part. There's a couple of them that you can take post infection, such as the rabies vaccine. As long as you get your first dose of that vaccine within 72 hours, you can be protected from the disease. But most vaccines you have to get prior to getting the infection for your acquired immunity. So chemotherapy, these are treatments that can be during a disease. So any treatment with chemicals is known as chemotherapy. This isn't just, it's a very broad term, let's put it that way. Chemotherapeutic agents are used to treat infectious disease, and these can be synthetic drugs or antibiotics. Antibiotics are chemicals that are produced by bacteria and fungi that inhibit or kill other microbes. Most of the antibiotics that we have today are derived from these. They're called natural product inhibitors. I did a lot of this research actually over in Oban in Scotland. That was really, really fun little stint. And what we were doing is we were searching for natural products that were derived from a marine environment. And I was doing these assays to see if they were actually, if they would be antifungal. And we did find a few that would inhibit. And then we sent them off to the chemists to find the exact structure, which is what you're looking at here. You see a number of different drugs and antibiotics that once we derive their structure, then we want to know what is the important part of an antibiotic, what is actually doing the job. And that's what you see in this, in the little purple boxes. And so when you make many different derivatives of an antibiotic, you might want to make it smaller. You might want to make it more efficient. You might want to make it bigger, actually, totally dependent on what you're trying to treat, how you're trying to treat it. But you're going to want to keep your little active group stable. And that's the group that you'd see there in purple. So these are natural products that we then take into the lab and use. Some of the first synthetic drugs include quinine. This was from tree bark. It still is. This tree still exists. And it was long used to treat malaria. Very interestingly, this is a tree bark that's down in South America. And when we talk about the disease malaria, and we will in later lectures, malaria is a super interesting disease. It's a parasite. And there's different types of that Plasmodium parasite. Falciparum, Plasmodium falciparum, and Plasmodium vivax. There's also a couple others, but those are the two main causes of malaria. Plasmodium falciparum we can easily trace to Africa, super easy. Plasmodium vivax is interesting. We don't know where it started. You know, it's very likely because there are monkeys in South America that it started in a monkey, and we don't know how monkeys got to South America, but it's presumed that they were essentially washed out to sea at some point on a big raft, and they made it over to South America, and then they populated there. And one of them may have taken malaria with them. But what's interesting about this is that the native people down in South America knew what malaria was easily. So you had the Spanish conquistadors and all these guys coming in, and they're like, this is a horrible disease you don't know anything about. And they're like, yeah, we do. Here's your drug. And so that was the first synthetic drug, quinine. That's kind of, it didn't happen exactly like the way I told it, but it's a neat story. So some other synthetic drugs. We have Ehrlich. He comes around, gosh, when was he in? Oh, the late 1800s. And he speculated about a magic bullet that could destroy a pathogen without harming the host. And then in 1910, he was able to develop a synthetic arsenic drug, salvarsan, to treat syphilis. Syphilis was pretty endemic throughout the world at that time, and they really wanted to get rid of it. And so what he did is he developed this drug. And if you look at the structure, which is on the left-hand side of our screen here, in the middle, that AS and double bond AS, there's your arsenic right there. That's your active group. Now, you can't have the arsenic on the outside of this chemical because it is a poison. And then it's not very stable, and it's just going to poison everything. But this particular one, the way that it's shaped, targets microbes. Let's put it that way. So it would target the syphilis bacterium. In 1930s, sulfonamides, so sulfa drugs, were first synthesized. And that's a structure that we're looking at on the right-hand side. We have the sulfur group in the middle. R stands for any chain. So there's an R1 chain, R2 chain, R3 chain. So you can change the structure of your sulfonamide dependent on its use, if you need to, say, cross the blood-brain barrier, if you need it to dissolve in the stomach. But with those chains, as long as you keep your little sulfur double bond oxygen active group there. So antibiotics, now while we had those drugs, it took quite a long time for antibiotics to really come into development. And this was really because of an accident. So in 1928, Alexander Fleming discovered the first antibiotic. And what he did is that he observed that penicillin fungus made an antibiotic, penicillin, and it was killing S. aureus. How did he discover this? Well, he was kind of a sloppy scientist, to be honest. And he had his bench had some shelves on it. And his wife packed fruit for him for lunch one day, and he didn't want to eat his fruit. So he left his lunch up there on the bench, and he never cleaned it up. So it was, I think it was an orange, because it's that type of fruit that usually does that. So it was sitting up there. Now, meanwhile, down on his lab bench, he was streaking out plates of S. aureus because he was doing some experiments on this microbe. And he noticed that his plates, so he'd streak them out, he'd then go and incubate them, and then be like, why? There's little dots on here. There's clearing dots. And then he happened to look up, and he saw a very moldy piece of fruit. And he thought, could something be coming from there? And sure enough, it was a penicillium fungus that was dropping down onto his plates and killing the bacterium. And so then he cultured that fungus from his fruit and was able to eventually make penicillin. And this was very important into World War II, when a lot of people were dying in the field again. And finally, we had an antibiotic to help treat them. And there we go. So in the 1940s, penicillin was tested clinically, and it began to be mass produced. And actually, during the war, I mean, there were literally bed pans. People would use bed pans to grow this fungus, and then they would strain out the penicillin from cheesecloth to treat people. And it worked. So modern developments in microbiology. So we have a few terms here. Bacteriology is the study of bacteria. Mycology is the study of fungi. And virology is the study of viruses. I'm just going to check our time here. We're doing all right. Knew this was going to be a long one today. I'm sorry, guys. We have at 10:50 we'll be done for sure. Parasitology is the study of protozoa and those parasitic worms that are disgusting and microbial life cycle. Immunology is the study of immunity. Vaccines and interferons are being investigated to prevent and cure viral diseases. So we have vaccines for viruses go way, way back. Now we're also trying to use some different techniques. Interferons are factors in our body that are used in the inflammatory response. We have 2 chapters on immunology, so we are going to talk more about this stuff later on. But using some anti-inflammatories or inflammatory factors that are naturally created in the body, we can mediate a viral response, immune response, sorry. The use of immunology to identify some bacteria according to serotypes was proposed by Rebecca Lansfield in 1933. So to start to characterize the diseases based on the immune response and the bacteria, that's really in the early 1900s we're able to start doing that. Recombinant DNA technology. We will have a whole chapter on this, but this is related to genetics, so microbial genetics. It's the study of how microbes inherit traits. Now we use that in molecular biology. So we can study how DNA directs protein synthesis. In genomics, we have the study of an organism's genes, and this has provided a lot of new tools for classifying microorganisms. Very often now, we don't classify an organism based on what it looks like. We classify it based on its ribosomal RNA because that's far more accurate. Now, recombinant DNA, in recombinant DNA, we have DNA from 2 sources. So using all of this knowledge, the microbial genetics, how microbes are able to inherit traits, how they switch genes around, the biology of this, how that DNA then makes a protein, and the genomics knowing the genome of a microbe, we can come out and start doing these recombinant biotechnological experiments. And we know the full genome of most microbes that we use in the lab. They've been fully sequenced. And if we don't know one, we go and we sequence it. It costs, I don't know, if you need to just find some species, what does that cost around? So I sent mine off for $400 and I had 6 samples. So that's, I mean, of the lake, I'm doing some work. I mean, it's pretty cheap. We can just do it. We also can sequence at SKC. We have sequencers. We're going to learn to use them. We just got them. So anyway, when you make this recombinant DNA, this picture I like, it's pretty nice. It's basically a cut and paste job. You take a gene of interest and you plop it into your DNA vector, and then you go on to express the protein.
Are we going to use those sequencers in this class?
No, unfortunately, our lab is going to be virtual this summer, and the person that we're hiring to do the sequencers probably won't be coming on until September. And then we'll have that going. But if you're really interested in it, you can always stop by our lab this fall and come in and see what we're doing. You don't really see a lot. I'm going to tell you right now. It's basically you just have this little cartridge and you pipette in your samples that look clear and you put it into a box and you close the box up and magic happens. It's not a magic box, but the sequencing happens. So you don't actually get to see anything. And then the data is transferred off to a big computer source and we get that back, and then we delve into a lot of data at that point. So no, cool. I'm sorry, not in this. It is cool. It is fun. I mean, yeah, you guys are always welcome to stop by our labs this fall when we're back open. So let's see. So in the 60s, this was the first time that we had people doing this recombinant DNA. And so this was Paul Berg. And he took an animal gene and put it into a bacterial vector. And then the bacteria reproduced, and he was able to find that that bacteria was making the protein of interest from that gene. And so that was the first time. And of course, now we do that all the time. And I mean, it's just falling off a log doing this stuff. We do it all the time. I don't think they have PCR on here, do they? Nope. PCR, I want to mention really quick. Polymerase chain reaction is when we amplify DNA, so we make more of it. And that was first, when did he do that? In the 80s, early 80s. And so when we started using PCR, now we're able to amplify our DNA. We're able to insert different genes. It was just a really rapid growth of DNA technology to arise where we are now, where we can sequence everything as well. It's just, yeah, pretty neat time. Microbial ecology. So we are going to have 2 big chapters on this later, but basically, microbial ecology is defined as when you have bacteria recycling carbon, nutrients, sulfur, and phosphorus. And then these can then be used by plants and animals. They don't have nitrogen on here, but I think they mean that through nutrients. The nitrogen path is also a really big one. And that one is really coming from a lot of organic decomposition. And then we have bacteria that live in the soil, and they're going to change the chemical makeup of these nitrates, or in this case, we're looking at phosphorus, in order for it to be used, to be taken up by plants, and then recycled in the atmosphere. So is that, nope, I guess I have a couple more slides. I don't know where this one ends. Again, all new stuff. I just made this PowerPoint on Saturday. So we have biological insecticides. These are microbes that are pathogenic to insects. So this is a really cool way to try and keep down these insects that are ravaging crops without having to use pesticides. So microbes are really they're used in many different ways. We use them to clean up oil spills. We use them to clean up lots of waste, but we can also use them to cause infection in insects, but they're harmless to other animals and to plants. Okay, biotechnology. I have talked about biotechnology briefly before, but it's the use of microbes to produce food and chemicals, and we use recombinant DNA. So this is this technique for biotechnology, and we can put genes into bacteria and fungi to produce any kind of protein or enzyme that we need, including different vaccines. Oh, here we are. And then gene therapy as well. So missing or defective genes in human cells can be replaced in gene therapy. Pretty controversial right now. I don't know if you've seen this in the news. We have CRISPR. CRISPR technology is a gene therapy that, well, it works. It definitely works, but it's not super specific, and in most of the world, it is not approved to be used in humans. But however, scientists in China went ahead and used it in 2 fetuses. They're twins, and he's been sanctioned. He's not working anymore. I don't know if he's behind bars, but he isn't around. But those 2 twins, what, they're 2 now or 3 now? They have found that actually their genomes, they were identical twins. Their genomes aren't identical anymore. So we'll just see how that continues. But anyway, CRISPR is a really interesting thing.
Is that what was,
oh yeah.
sorry. Was
Go
that
ahead.
what was being used for sickle cell?
No, we don't use CRISPR in sickle cell. Like I said, it's not approved to be used for humans. CRISPR technology right now is still being studied and investigated. It's mainly going to be used in bacteria, in fungi, in genome sequencing, in different biotechnological uses, but it's not going to be directly put into humans. There are some people that are trying to do that, but because it's not super specific, it can jump and it can change and it can get into the introns, the interruption of genes. It's changing the way things are expressed. There's too many unknowns, let me put it that way.
Yeah,
So
it sounds
it's
awesome and terrifying.
exactly, that's exactly how it should be summed up. It's awesome and terrifying. Don't use it yet, but in the future, once we really understand it, it could be great. But it should remain in the lab for now. And some people are trying to jump the gun on that, obviously.
Thank you.
Yeah, no problem. Oh, but we can use CRISPR, so that goes on to this next bullet point, that we can protect crops from insects and from freezing. So you can use CRISPR in GMO for plants. And we can do that because, well, we don't always. If you really want to target a gene, say, for example, they had a wheat in the UK, and they said, we want to put a hops gene into this wheat. You know, hops is found in beer, right? Well, hops also has this really, has a smell to it, and it turns out that aphids really hate it. So if they put just this one little specific gene into the wheat, then they wouldn't have the aphids eating the wheat. And so it's just one very specific plant gene being put into another plant. And it's a very mild change. And that one you wouldn't want to use CRISPR. But CRISPR has been used in some different plant GMOs. One of the really cool GMOs that's out there right now is the grass that they're growing in the Sahara Desert. Oh yeah, that's neat. So by using biotechnology, and I don't believe they actually used CRISPR for this one because it's been done for a little while now, they're able to create a grass that can literally grow in sands, in big giant sands. And as you may or may not know, the Sahara Desert is growing and it's taking over whole villages, and it's not a nice place to live, the Sahara Desert. And so by being able to introduce grass, they're able to push back against those sands and start to reclaim some of the land. And bring back water and cool that area down. So to me, that's a super interesting one. Let's see. Okay, normal microbiota. So microbes that we have in and on our body are called normal microbiota. So we are colonized. We are just, yeah. So whenever people come into the lab, I don't work with anything that's dangerous. But I have people wear gloves and a lab coat if you're going to go into tissue culture, and you have to work in a hood, and I might, say, keep your hands in there, that sort of thing. And we use ethanol to spray things down. I'm not doing that to protect you. I'm doing that to protect our experiment, because I don't want what's on you to get into our beautiful tissue culture. I don't want to be another Alexander Fleming, although that was a great accident, where the fungi dripped onto the plates. We don't want to do that. And generally, when you infect your experiment, it's not going to go well. But normal microbiota, oops, I thought I had one more sentence there. Normal microbiota are on your skin, in your body. They're there to protect you. They keep you colonized so that pathogens can't colonize you. So if you get a cold, like I've got a little cold now, I'm just going to drink a lot of water and get on with it. But if I were to take antibiotics, I would then want to eat a bunch of yogurt, because I don't want to get rid of all my normal microbiota, because then I might get a secondary infection. And then that's super fun. Now I've got a gastrointestinal infection along with the cold. The normal microbiota on your hands is fantastic. I mean, you can kill a lot of things just by touching it. So They lower the pH on your skin. They keep out those pathogens. They're part of your immune system, really. Biofilms. So biofilms are when we have microbes attached to solid surfaces, and they'll grow into masses. They can grow on rocks, pipes, teeth, and medical implants. Anything that is not natural into your body, a medical implant, can grow a biofilm. So we really have to be very sterile with these implants. And of course, your teeth. I did brush my teeth. I was just going, rub your tongue over your teeth. You'll go, oh, there's a biofilm starting. I should brush my teeth better. When you need to brush your teeth, you feel a biofilm. Let's see. Infectious diseases. This is when a pathogen will overcome your resistance, the host's resistance, and we result in a disease. Emerging infectious diseases are new diseases or diseases increasing in incidence. Down at Hamilton Labs outside of Missoula, this is what they do. They really focus on emerging infectious diseases. They are one of only a handful of CDC labs that do this. Why it's in Montana? Probably they thought nobody lives there anyway. Put the really dangerous stuff there. All right, so that's the end. So in summary, microbes include life forms that are too small to be seen by the unaided eye. We include animal parasites because they have a microbial stage of life. There are 3 domains of life: bacteria, archaea, eukarya. Eukarya do have some microbes, protista, fungi. And I wanted to put this last take-home message here that most microbes are not pathogenic. I really want to stress that because we do have all this biotechnology, we do have the normal microbiota, but we are going to focus a lot on the pathogenic microbes, and that's because we're human, and that's what we care about. We look at somebody who's sick and we want to make them better. But most microbes are not pathogenic. So I'm going to escape out of here, stop presenting, and I'm also going to stop recording, okay, so that maybe I can stop recording. Hopefully this will upload.